viewer. Today we want to continue with growth and development, and today we are moving to another aspect of growth, and we'll be looking at measurement of growth. Now, when we want to look at measurement of growth, there are various aspects of growth that we need to know that we will be looking at for us to determine growth in living organisms. Now, the aspects that we have within living organisms include height, volume, mass, and even length. So you can always find that we can always plot a graph of the growth parameter against time, and from there we can be in a position to estimate growth that is taking place within the body of an organism. Now, when we always plot a graph of the various parameters of growth against time, we'll always obtain a curve that that is always an S shaped, and we refer to it as sigmoid curve. Now, if you look at this diagram we have here, it is showing the growth that is taking place within an organism. We can have any parameter, e.g. height against time. Now, growth always takes place in different phases, and we are going to look at the first phase. So the first phase, we always refer to it as lag phase, or what we can call it slow growth. We have a lag phase. So we always have a slow growth. So what happens during this phase is that very little growth always occurs, and this may be due to the following. So the growth rate is very slow because the number of cells which are dividing are few. Then another thing is that the cells have not yet adjusted to the surrounding environmental factors. So if you look at this graph, we have where we have small a, you realize that we have that lag phase, and we've said that the growth is very slow, and this is due to few dividing cells, and cells have not, the dividing cells have not adjusted to the surrounding environment. Now we move on to another phase, and that is exponential phase, or what we call log phase. Exponential phase. Now, when we look at this type of phase, it is the second phase in which growth is always rapid, and it will always proceed exponentially. Now, during this phase, the rate of growth is always at its maximum, and at any point, the rate of growth is always proportional to the amount of material or number of cells of the organisms, to the number of cells of the number of cells of the organisms that are already present. Now you find that in this phase, growth is very rapid because of the following. Number one, there is an increase in the number of cells which are dividing, and the number increases geometrically. Now, for example, when the cell divides into two, then later this other cell will divide into 4, then 8, then we can have 16, and so on and so on. Another reason why we have a high growth rate or rapid growth rate is that the cells have already adjusted to the surrounding environment. And another thing is that food and other factors are not limiting. So you find that cells do not compete for these resources because the resources are abundant. Now, lastly, we have this phase being rapid because the rate of cell increase is higher than the rate of cell death. Then that one will make the rate of growth to be higher. Thirdly, we have a decelerating phase, and this is the third phase where growth takes place. Now, during this phase, you realize that growth becomes limited, and as a result of the effect of some of the internal factors or external factors, or a combination of both the internal factors and the external factors. Now you find that growth will be slow at decelerating phase because one, fewer cells are still dividing. Then another thing, we have the environmental factors, that is the internal and the external environmental factors, such as shortage of oxygen and nutrients due to high demand by the increased number of cells. Because in the exponential phase, we had mentioned that the cells were increasing. Now, because we have a high number of cells, there will be competition for these resources. Now, another factor, we can have space. Space is always limited due to, again, high number of cells. Another thing, we have accumulation of metabolic waste products that will always inhibit growth. We also have limited acquisition of carbon dioxide, as in case of the plants. Then lastly, another cause of slow growth rate is the fact that most cells are fully differentiated. Now we move to the last phase, that is we have plateau, and that is the fourth phase, or we can refer to it as stationary phase. Now this is the phase which will always mark the period where overall growth has stopped or it has ceased. So you realize that the parameters that are always under consideration will always remain constant, and this will be due to the fact that the rate of cell division is always equal to the rate of cell death. Another thing is that nearly all cells and tissues are fully differentiated, and therefore there is no further increase in the number of cells. Now, from what we have learned, you are in a position to note that in the lab phase we had slow growth rate, and this was because of few dividing cells and other reasons. Then we move to this other phase, which is the second phase, that is the log phase or the exponential phase, where growth was rapid, or growth was proceeding exponentially. Then the third phase we had where we have C, decelerating phase, and we look at the reason why the cells were the growth of cells was limited, and this might be because of the cells which are dividing are few. Then lastly, we looked at the plateau phase or the stationary phase, and in that place, if you look at the parameters that are under consideration, they always remain constant. Now you realize that the nature of the curve during the phase of plateau, you will come to realize that they will always vary. The nature of the plateau phase will always vary depending on the nature of parameters that are in questions. Now, you realize that another factor which can always make the nature of the last phase to vary, maybe the species that is being studied, and even the internal factors. So you realize that in some cases, the curve may always continue to increase slightly until the organism dies, as in the case of monocotyledonous plants. But we can also have the same case taking place in many invertebrates. We can also have the same case in fish. We can also have the same case in certain reptiles. Then in such a case, we talk about, or when we have such kind of a scenario, we refer to it as positive growth. It will be indicating positive growth. Now, in another case, you can always find that the curve will always flatten out, and when it flattens out, that will indicate, will be indicating that that there is no change in growth, while other growth curve, the other case, actually the last case, it may tail off, and in that case we'll be having a period of negative growth rate. Now, this negative pattern is a characteristics of many mammals, including human beings, whereby you find that physical senescence is always associated with increasing age. So this is the last plateau. So we say that at some point we can always have it increasing, or we have it tailing, moving upwards, or it can flatten, or in some case it can tail off. So we've given an example. We have human beings. Now, if we look at growth, this is always not the case of, in this kind of curve will always not give us all the growth, how growth takes place in all living organisms. Now, you find that when we look at sigmoid curve, actually does not apply to all living organisms. For example, we have the arthropods, and we will be looking at the insects. Now you realize that some parts of the body of the insects, like the thorax, is always covered with the, it is always covered with the exoskeleton. So what happens is that if volume changes within, that is the growth parameter. Actually, I'm referring to this diagram. If the parameter which in question, let's say it is volume, if volume changes are plotted against time, a different curve will always be obtained. We will not get the same curve as that one that we found for the case of sigmoid curve. Now, this kind of curve that we will always obtain, we will refer to it as intermittent growth curve. Now, in intermittent growth curve, and we refer to insect, the intermittent growth in insects is always due to the fact that they have an exoskeleton. Now, what happens is that before this exoskeleton is shed off, in the process of molting or echidiasis, growth can always not take place. However, what happens? The cells can continue to divide, but there will be no expansion because of the exoskeleton which is in place, and that one makes the space to be limited. So in an area where we have the unshed exoskeleton, the cells will always divide, but there will be no expansion of the cells due to limited space as a result of the unshed exoskeleton. So we have a practical activity whereby we can determine the measure of growth of a plant. So these are the things that you require. You require small pots or boxes, a meter rule, and seeds of beans. So procedure, you place soil in the boxes or prepare a pot outside the laboratory. Then you plant the seeds in the box and place it in a suitable place outside the laboratory. Now you water the seeds daily. Then you make observation daily until the seedlings start to emerge out of the soil. Then after that, you measure the height of the shoot from the soil level up to the tip of the shoot. Then you repeat this for the other seedlings. Then you come out with the average height of all the shoots for that particular day. Then you repeat the same procedure, that is procedure 5, after 3 days for at least a period of 3 weeks. So then after that you record the results in a table form. Then on the same seedling, measure the length of one leaf from each of the 5 seedlings. Then you calculate the average length of the leaves and record in the table. Then from that you can plot a graph of the height of the shoot against time. So another thing, you can on the same axis, you can always plot the length of the leaf against time. Then you compare the two graphs. So from today we've learned about 2 types of growth curves, and the first one we've mentioned that it is a sigmoid growth curve. And another type of growth curve that we've learned about is intermittent. Intermittent growth curve. So you realize that in intermittent growth curve, mainly we find it in arthropods, and we've used an example of the insects. So you can look at what we have within this graph. You find that we have within the intermost, this is where we have molting taking place. So in this case, this is where we said that the cells can still divide, but because there is presence of the exoskeleton, the cells we cannot have the expansion of the cells. But in this area, we have the growth phase. So after this, you find that after this point, you find that the exoskeleton has been shed off in the process of molting or echidiasis. So from here, growth can always take place in this part. So it continues until the insects reach the adult stage. We hope to meet you in the next lesson as we continue to learn about growth and development. So have a nice time. Remember to subscribe and to comment, and any question will be answered. Thank you.